All right, we're ready to go. And for those of you who haven't heard, Dr. Villa had a family emergency. She can't be here today, so it's gonna be real, and Dr. McCullough can't make it today, so we're real informal and ask questions and just enjoy yourselves, and we may get out of here a little early.
Yeah, I definitely want to take an hour. So.
Okay. All right.
Cool. So I'll talk today about one of my current projects that I'm calling multi-model modeling. And the talk kind of has some applications to blood glucose and insulin measurements in diabetes. So, and I guess I'm Nate. I'm in mechanical engineering. So the problem we want to address is that we use mathematical models to study all sorts of biological systems, but in practice, we don't usually know the correct model to match the biology that we want to study. So we can come up with a formulation and it maybe approximates it, and we could make a different assumptions and come up with another model formulation. And we can imagine doing this for as many assumptions as we can come up with about our system. So we'll have a whole family of many models, and we really don't know which is the right one and which one we should use in application. So today I'll be talking about kind of one story of multiple models, and that's making diabetes diagnostics in the clinic. And we do this with all sorts of different clinical tests, one of which is the oral glucose tolerance test. So the patient has fasted prior to this test, they go into the clinic, they drink a bolus of glucose, so 75 milligrams, or like a drink with sugar in it. And then that'll spike their blood glucose, and we can measure the response of insulin, another molecule called C-peptide, which is secreted in a 1-to-1 ratio with insulin, and it's easier to measure, and then glucose in the blood. And then we can use this data to make some sort of prediction and some sort of diagnostic about the patient. And these predictions, these diagnostics are made using mathematical models. So in this setting, we can come up with a family of 3 different models, which are all based on the same kind of simple transport of C-peptide into the bloodstream, C-peptide out, and then some response function, which is this the bottom line in all 3 blocks of how the C-peptide is secreted with changes in blood glucose. So the 3 models make different assumptions about how the pancreas responds to changes in blood glucose level. And then we can use the models to come up with some sort of indices or numbers about the patient that we can use to make a diagnostic. And these things are actually used in the clinic to make diagnoses about patients. The problem is we have multiple models, we don't know which is the right one, and we want a way to reconcile all of the predictions from the multiple models at the same time. So that way we're not biasing our assessment of the patient on one specific model formulation. So kind of our intuition will tell us, okay, let's figure out a way to pick the single best model, and we'll run with that. So we can have predictions, and then we could do some sort of ranking and model selection to rank the models and get our best model, our worst model, and a mediocre model. There's all sorts of tools to do this, so I'll run through a couple of them. The first one is this these information criterion. So we want to kind of understand how much information in the data is being captured by the model predictions. So the AIC essentially tells us just that. It says we want to look at this loss function, which is the log of the likelihood function, which kind of measures the mismatch between the data and the model predictions at the parameters of the model that maximize this, and then it's balanced by the number of parameters in the model. From a very, very complex model with many parameters, think a polynomial order 100 is going to fit the data perfectly, but it's way too complex. So that'll penalize the score for having a lot of parameters. A very simple model is not going to capture as much of the information in the data, but it will penalize less for having fewer parameters. And then we can do this in a Bayesian way by computing Bayes factors. So we want to look at the probability of the data set under a specific model. So that's this probability of data given model, and then we'll compute ratios of that for different models. And we'll be able to rank models based on these ratios of probability. So in reality, we're going to want to work with all of them. instead of just picking a single best one. Why? Because maybe the best one will be overfit to the data by still being simple, and then we try to go take different data and it doesn't do as well. The true data generating process might be a mixture of these. So one of them is a linear response, the other is a logarithmic response, maybe it's a linear and a logarithmic response. Or maybe it changes in healthy patients and sick patients, and we don't want to bake in those assumptions at the start. And then there's a whole bunch of theory about when you average over multiple model predictions, we'll get more robust predictions. You'll get higher certainty, you'll have reduced variance when you do these averaging type approaches that we'll talk about. So we want to use multiple models to improve the robustness of our predictions, and so we don't bias towards specific model formulations. But how do we combine them? You could think of just averaging across things. That's probably not the smartest way. So we're going to talk for the rest of the talk about how we actually combine model predictions. So before I get into that, I want to set the scene and just build the mathematical framework of this modeling with multiple models. So we assume we have K computational models of the same biological process. So here a computational model is a differential equation, or it could be a neural network. It could just be like a linear regression model. This framework is general enough, it doesn't care about the flavor of the model. Each model maps from some parameter space to some output space. So that's from model parameters to predictions of insulin sensitivity, or these metrics that we're going to use to make a diagnostic. And then we can define some quantities of interest in that output space. So further, rather than assuming the model makes a single point estimate for these diagnostics, we're going to assume everything is probabilistic and the predictions are come as a probability distribution for these output quantities. This happens because we calibrate the models to the data using Bayesian inference. And if you've seen my previous talks, I talked all about Bayesian inference, but it boils down to applying Bayes' rule to calibrate model MI to the data. So Bayes' rule tells us that the probability of the model parameters conditioned on the data set we have and the model formulation is proportional to some assumptions we make about the parameters before we look at any data, and this likelihood function, which is a mismatch function. So if the data is really bad, this probability will be low, or the model predictions are really bad, that probability will be low, and if we have a really good prediction using a set of parameters, so that probability will be high. So essentially what we'll get is we'll get a collection of samples of the distribution of the quantity of interest under a specific model. We'll have different distributions for the different models. So the simplest way of fusing probability distribution is what's called an opinion pool, and this goes back to the 70s and 80s. So we want to construct this consensus distribution, so the probability of the quantity of interest conditioned on the whole family of models, all the data, and all the parameters of those models. So we can do this by just choosing a set of weights for each model, and then the consensus distribution is just a weighted average of each distribution given each model. But how do we choose the weights? So this theory just says you have some weights, they're probably just given to you. But how do we choose them? So we're going to talk about ways to choose these weights. So the simplest approach we can think of is we'll model the whole thing parametrically. So our consensus distribution will now be a parametric distribution. The parametric distribution is something like a Gaussian distribution. So that has parameters mean and variance that control the shape of how that Gaussian looks. We can do that in such a way where we compute the mean and variance of our consensus distribution from the means and variances of all the distributions we have from our models. And that's what this Winkler 1981 approach does. So our consensus distribution is this new distribution, and it has mean and variance that come from all of the means and all of the variances of each model that goes into it. And this is good because it can handle correlations between the models. So maybe the model formulations were very similar, so they'll be correlated. It's simple, but it only works for scalar quantities of interest. So let's assume we have a dynamic quantity of interest. This approach won't work for that. And it only weights by predictive uncertainty. So you can kind of get a sense of the weighting comes from these inverse of this covariance matrix. So the covariance matrix has the variances of the predictions from each model. And it's only weighting, so a model that's got a very large variance, very big number in that matrix, will have a very small weight. And a model that has a very small variance, it's a very, like it predicts the same value all the time, will get a really big weight. But maybe that model doesn't agree with the data very much. Even though it's predicting with high certainty, it doesn't agree with the data, so it'll get misweighted in this framework. So then we can think of weighting based on information criterion. So these are the AIC numbers that I talked about before. We can construct weights using them. These are good because they weight by how well we match the data. However, these standard information criterion only use a single estimate for the parameter, so it's not doing a whole probabilistic thing. So we can do it in a Bayesian way where we instead either do Bayesian model averaging, so we use the Bayes factors that I talked about before as the weights, or we do what's called pseudo Bayesian model averaging, where we're going to construct something that looks like an information criterion from the whole posterior distribution, so the whole distribution of each model, and we'll use that to assign weights. And I'm not going to get into the details here, because it gets tricky if this isn't the stuff you work on all the time. I'll talk about two methods in the application. So the Winkler method, and we'll kind of look at how it fails, and then we'll look at the pseudo Bayesian model averaging approach, which is kind of the state of the art that people like to do. So as a first example, I wanted something where we know what the true model is, and then I could come up with a family of other models that agree or disagree with this true model, and then we can see how they're being weighted and accounted for in the average. So this is a true model. It looks kind of like a Michaelis-Menten formulation of enzyme kinetics. It predicts the kinetics of an enzyme catalyzed reaction with changing substrate concentration. And then as data for the problem, I took 4 noisy samples from this. So that will be the data that I use to calibrate all the subsequent models. And then I constructed a family of 3 other models. So this is some reduced model. It assumes that some of these parameters go to 0. And then I can fit similar behavior with an exponential function and a sigmoid function. So these will be the models that we want to average over. I calibrate them to this data. So I do the whole Bayesian inference that involves setting up the whole Bayesian model and then sampling from that posterior distribution using Markov chain Monte Carlo algorithms. And I get distributions out for the predicted trajectories from each model. So that's what we see here. So this is the error bars are 95% of all the simulated evaluations of the model fall within these shaded bands. Then I can construct a quantity of interest from this. So I'll just look at the steady state value and I get distributions for the steady state value under each model. Now we want to combine all of these distributions into a consensus distribution. So if we use the Winkler approach, we see something that looks like this. So the 4 original distributions are in these dotted lines, and then the consensus distribution is this spiky distribution that's in the middle. And we're thinking, of, okay, that looks nothing like the true steady state value. The true values are around 1, which is what was predicted by the true model originally. Why is this pulled all the way to the left like it is? And that's because of this reduced model has a much smaller variance than these other models. So that's going to get a much stronger weight than these models that are actually doing better than this reduced model. So we don't like this. This is telling us that this method doesn't really work in the setting. Another thing that's nice to look at here is you can see how pointy the consensus distribution is. So you can actually prove that when you do this averaging, you're going to reduce the variance from your original models to the consensus model. So the variance of the consensus distribution will always be lower than the variance of all your models together. So that's nice. We're guaranteed to have a more robust predictor when we do this. But okay, so what are we going to do instead? We'll look at the pseudo Bayesian model averaging approach. And plotting two different methods for computing this. One of them is this widely applicable information criterion, and another is a leave one out cross-validation estimator for the same underlying quantity. And we can see that both of them lie right on top of each other, and they agree very nicely with the three models that approximate the right behavior. So the mean is close to 1, but the variance now is tighter than all three of those models that we saw before. So now we have a way to weight the models based on how well they agree with the data, and we're guaranteed to find the variance is going to be smaller. We can use the same weights now to get predicted trajectories. So we just run a whole bunch of model simulations and then take a weighted average of those simulated trajectories using the same weights that we used to compute the weighted distributions, and we get these gray trajectories which perfectly cover the data and agree very nicely with the original true model. So now we're going to turn back to the beta cell insulin problem, and we'll look at a couple of results using those models. So, like I said, our goal of applying these models is to take patient data that looks like this and predict some sort of quantity of interest that we can use to make a diagnosis. So here I'm going to give this patient data. So these models take glucose measurement as an input. So I interpolate over them, and then I have a continuous input to the model, and then I fit them, the predictions, to this C-peptide molecule, which is secreted 1-to-1 with insulin. And then from the model predictions, I can compute this beta cell glucose sensitivity index, which is the area under the curve of predicted C-peptide normalized by some area under the curve of glucose above predicted baseline. So back to the three models really quickly. We have this Havorka 1998 model, this Creddy 2001 model, and the Breda 2001 model. And the three models differ in how they assume that C-peptide is secreted in response to changing glucose. So there's kind of three different formulations for this input function. And they increase kind of in complexity. So this is linear. This one assumes that there's some hidden variable that evolves with some dynamics. And then this one has another hidden variable that evolves with different dynamics that depend on both the current glucose level and how much the glucose level is changing. So the first thing we're going to do is we'll predict, again, some of these model parameters from the data. And we do that, we get a whole set of predicted trajectories for the different plasma C-peptide dynamics. So on top, we have the three models for the healthy data. And then on the bottom, we have the three models for the data from patients with diabetes. And we can see that some of the models agree better than the others. So this Creddy et al. Model, it doesn't predict the end very well, and it kind of misses the initial transient, but it gets the top very well. Similar story with the Havorka model, and then the Breda model underestimates at the end. So then we can compute the area under the curve of these C-peptide predictions and get distributions out for area under the curve. So for today, we're just going to talk about area under the curve and not the full sensitivity index. So we see that for the patients with diabetes, the three models predict similar distributions for C-peptide area under the curve. However, for the healthy patients, there's kind of a bigger spread, with Breda underestimating compared to the two other models. So we'll apply both of our mixing methods to this problem, and we'll see kind of what happens. And it's going to be a similar story to the enzyme kinetics example, where the Winkler method is biased towards kind of the average of all three weighted by the variances, whereas the pseudo Bayesian model averaging weights by how well we agree with the data. So in this bottom in the top plot, we have the results from WINKLER, so the diabetes patients kind of make sense, it's a tighter average of all three, and then this healthy one is kind of biased towards this underestimating model, whereas with the pseudo-Bayesian model averaging, we have very similar result for the diabetes patients, but in the healthy case, we're much closer to the average of the models that agree with the data by eyeball more, and we're not biased towards this model that underestimates. And then we can imagine from here using this to construct some sort of classifier, or we could take a new set of patients, predict scores for them, and use these distributions to classify whether they have diabetes or not. So kind of what's next for this work is further applications. There's many other tests, many other models that we want to talk more about what's useful in the clinic. We're going to work with some clinical collaborators who collect data here at UCSD from patients and use it to actually help them with predictions in the clinic. So inclusion of actual patient data and then some population modeling. And then there's other fusion methods. So I didn't do any true Bayesian model averaging. There's some computational challenges there that the community worries about. And then there's other things called model stacking that we could talk about. And then I didn't talk about time-dependent approaches. So all these approaches assign a single scalar weight to the model, and when we make a time-dependent prediction, we just weight everywhere in time the same for that model. But you could think, okay, one model maybe gets the beginning part right, and another model gets the end part right. Maybe we want to weight that changes over time. So there's approaches such as dynamical model averaging or the sequential data estimation, where they're learning a dynamic weight. So a whole bunch of weights over the whole time that you want to predict that allow which models doing better to change at different parts over time. So that's where I'm at now, but thanks everyone for listening, and I'm happy to go through anything else in more detail. Yeah.
I missed the first 10 minutes, I'm sorry.
It's okay.
But would you consider expanding this model to like a whole umbrella of dysfunctional diseases that are induced by high glucose, high insulin resistant states? Because we know that high glucose doesn't just drive type 2 diabetes, and high insulin doesn't just drive type 2 diabetes either. There's a differential response, and that some people who have these conditions for years before they even develop type 2 diabetes are already changing like the beta amyloid plaques in the brain, could be leading to PCOS in other patients. So do you see in further applications of these models like an umbrella of like just Western diet induced diseases that are driven by these chronic exposures to glucose or high insulin?
Yeah, absolutely. I think the challenge there is the biology is still emerging, so we're just learning the right things to model in those settings. Like we don't know how glucose receptors or on different parts of the body actually affect function. So for me right now, I'm just taking models that have already been written in the literature. I'm not writing any models myself for this work. But we were thinking about studying obesity and trying to come up with models for obesity, so on and so forth. So I think you can definitely do it. It's kind of beyond the scope of the questions I'm asking. But
I have a question to jump off of that.
yeah.
I remember before you were looking into like AMPK proteins, which obviously have a lot to do with type 2 diabetes. Like, how do you decide, I guess, what you want to apply your models to? Like, how did you decide for this project? Was it like, oh, this is in the literature, like there's 3 and 3 is good enough, or 3 is a lot? I'm not sure because I don't read like the math literature, but
So we picked a disease we wanted to work on. And instead of looking at a molecular scale here, so the model's not saying AMPK signaling. I'm actually working on another project with Professor June Dog on AMPK signaling.
okay.
But here we picked a higher scale, so we want a model of glucose insulin dynamics over the whole body. It doesn't consider individual molecules or very small scale interaction. What specific clinical test has many mathematical models associated with it? And it turns out for the past 3 or 4 decades, people have been using models to make these types of predictions in diabetes.
And then would you say like those predictions are not very good and that's why you guys are tackling?
Right, these predictions are all based on assuming a single model. They don't tackle this idea of model uncertainty. This is one way to bake in model uncertainty into the prediction.
Another quick question, which maybe is a little foolish when I just missed it, but in terms of when you're comparing different models and you have the kind of error bars or the 95% confidence intervals, so that's stemming from you're doing kind of a Monte Carlo selection of the parameters of the model to fit the data, or how are you generating those distributions and eventually fitting the data?
So I do Markov chain Monte Carlo estimation to estimate the model parameters. So what that does is I'm going to start sampling from some distribution, and then I'll accept or reject parameter combinate, like set the parameters based on their posterior probability. So if they have a high posterior probability, I'll accept them, and then those will help me in my random walk or sample later. And eventually, after a long time, I'm sampling directly from this distribution.
Okay.
And then I can say, okay, I've generated 10,000 samples from that distribution, then I'll simulate the model with those 10,000 distributions. So I get 10,000 simulations, and I can compute. These are actually called credible intervals. So the lower bound is the 2.5th percentile, and the upper bound is the 97.5 percentile. And these are nicer than confidence intervals because we can say 95% of our predictions lie within this band. It doesn't have the weirdness of interpreting a confidence interval.
Sure, and that makes sense. So I guess that process of fitting the data, if you're doing tens of thousands of iterations, that's probably that's where most of your computational time is, right? So like how long would it take you to generate?
So these models are pretty fast.
Okay.
I think I'm doing like a minute and a half.
Oh, okay.
For the MCMC, depending on your model. So like people down in SAO work with weathered models.
Yeah,
You can't do that
no.
for this. So there's other techniques where you get approximate samples from the posterior distribution, so on and so forth. But it's usually on the time scale of hours to days.
Okay. So if you had like a model with say like, I don't know, 40 parameters or something, that'd be a while to.
Yeah.
Okay.
Larger parameter space is also trickier to sample.
Yeah, of course.
Not just from computational time of simulating the model.
Right.
So that extends the runtime as well.
So you chose 4 models here to represent the mixed one. How do you know what's the right number? Like, can you just go on? Does it converge to something?
It depends on the problem. So certain types of model families can be written such that like there's an infinite number of possible models, like maybe a graph where you have an infinite number of possible connections or many possible connections, and you don't know what the right connectivity is or something. In this case, that's a really tough question. Really, I'm looking at it, we have a bunch of models based on different assumptions. The number depends on the problem, how much we've studied it, what the different possible assumptions are, what the different disagreements in the biological literature are,
Right.
so on and so forth. I don't think there's convergence guaranteed, like if you have an infinite number of models, you'll converge to something. But I would look at it as you want every model with every assumption, unless you know it's completely wrong. This is kind of a nice toy example where we know the reduced model is completely wrong and the methods are agreeing with us.
So basically, whatever model that's been verified in literature, you can just pick up and go on and work with it.
Yeah. As long as it predicts the data and the quantities that you care about. Cool.
Questions? No? Okay. Thank you so much, Nate.
Yeah.
Good job. So can,